Hello, everybody. My name is Jeremieh Hasley, a University of Hawaii PhD student in tropical plant pathology and a tropical agriculture Ellen M. Koenig ARCS Award recipient. Throughout my studies of genetics, a beautiful observation about genes and evolution is that there are many connections between different organisms. And with these connections, we're able to understand a gene in one organism and then identify and infer its function in another. However, there are many economically important crops that have a relatively longer life cycle, ultimately meaning that the pace of plant science research is often limited by the rate of plant growth. And a way for us to overcome this is by the use of model organisms. Since they typically have a shorter life cycle and a relatively smaller genome of lesser complexity, we're able to take the information gained from that model organism and then implement it into other economically important crops. So for my research, I'm further developing the foundation to make papaya into a model organism by developing a precise gene editing system using the CRISPR technology. And in this foundational system, I'm targeting the commonly used proof of concept phytoene desaturase gene that if edited properly, an albino phenotype is expected. And while I'm underneath this genetic hood, I'm aiming to increase the disease resistance of papaya to a fungal pathogen by targeting the MLO6 gene. Now, an important aspect of this entire project is ensuring that our gene editing components, such as our single guide RNA and our Cas9 protein, are being properly expressed. Because if they're not properly expressed and properly complexing, then we don't expect to see any edits within our genome and target region. However, by using our developed gene editing system and then extracting the potentially edited genomic DNA and then having it undergo some deep sequencing, when looking at the target region, we're actually able to see edits within that target region, indicating the early success of our gene editing system. Furthermore, we're able to remove these gene editing components, leaving only the precisely delivered gene edits. So developing papaya as a model organism will rapidly advance the field of genomic research of slow-growing fruit tree crops such as oranges and apples, all with the aim to help researchers and farmers to create crops with better agronomic traits such as fruit size, taste, and even disease resistance. With the development of this system, you may see a wider selection of fruits, maybe even some with a longer shelf life, all the while reducing environmental impacts. And it could be coming to your local grocery store sooner than you know it, only after proper federal safety testing procedures, of course. Thank you.